action, and what I really like to do is clinical trials, and that's why it sometimes seems that I am so unfocused and doing so many things. But I like to work with participants and patients. And that's my interest. So if we're going to talk about cure of HIV, let's start with some definitions. And in the area of cure, people talk about sterilizing cure, which is basically no detectable infectious virus when you look for the HIV virus, or sustained remission. So what it means is controlling viremia in the absence of antiretroviral drugs, but you still can detect the virus in the blood or in the PBMCs or in the memory cell compartment of individuals with HIV. Because your understanding of what constitutes a cure is important, because when you are looking at clinical trials and you're trying to ask the question, are we there? Like the kids ask when they drive a car. If the goal is to do a sterilizing cure, then the endpoints of trials should be measure the size of the latent reservoir and not being able to detect the virus there. If you want to control in the absence of antiretroviral, you have to stop antiretrovirals and see what happens. So depending on what your understanding is or what your goal is in a particular study, that's your definition. There has been only one sterilizing cure of HIV. Timothy Brown, in this picture, had a leukemia, was transplanted, everybody knows this story, transplanted a bone marrow from somebody that didn't have the co-receptor for HIV, and he needed 2 transplants and a very aggressive chemotherapy, and he developed graft versus host disease, but antiretroviral therapy was stopped and the virus didn't come back, and most of the studies that have been done on him, and he has been a real advocate for cure research, has been negative. So there is no trace of HIV in his body, which I think also is important because it tells you that the reservoir has to do something with the marrow, with the cells, and that probably there is not many significant other reservoirs, or at least you need the CD4 cells to expand the infection. This is a barrier for HIV cure, and I've always put this slide for students to explain the problem for the immune system. So this is an Olympic swimming pool in China that has 1 million balls, and they have 2 colors, but imagine that all of them look the same, and only one has the HIV virus, only one has the prize, and the immune system has to find that ball among 1 million, 1 per million memory CD4 cells carries replication competent virus. And the balls look exactly the same to the immune system from the outside because the virus is sitting there laterally, not expressing any proteins. So it's not an easy problem to solve. You want to eliminate the reservoir without killing the individual or killing the other balls. And to find them is the big problem. There are measures, different measures different ways to measure the reservoir. So people are looking in what is perceived as the main reservoir, which is the resting memory T cell, and you can look at integrated HIV DNA. You can look at total HIV DNA, which is the addition of integrated, so virus, provirus that is inside the DNA of the patient, LTR circles that are in the cytoplasm of linear DNA that is the proviral DNA. And that the addition of all those 3 compartments gives you the total DNA. You can measure the integrated DNA, and there are assays for all these. And some people think that also the level of viremia, what is called the low-level viremia, correlates with the size of the reservoir, because these cells that carry HIV are always having some level of expression and replication, ongoing budding of the virus, and that this represents correlates with the size of the reservoir. The problems with all these measurements is that you can also measure HIV-associated RNA in CD4 T cells, is they don't correlate to one another. So when we are measuring the reservoir, depending on the method that you use, you get a number, and they don't correlate to one another. Most people, sure. Yeah, I know, this is not the p-value. Yes, this is an old slide, should be corrected. But the point of this is, yeah. So the point that Una is making for the audience, they didn't hear the question, is that there is a typo on this slide, and because I took it from an old slide deck, that there is no correlation. The integrated DNA measure correlates with the viral outgrowth assay. So, but the point of this slide is that you get different numbers depending on the method that you use, and they don't correlate to one another. The gold standard is because you want to make sure that you're sequencing virus that is replication competent, has always been the viral outgrowth assay. So the frequency of this, even if you have chronic infection or acute HIV infection, there is less reservoir if you measure the reservoir in an acute infection, is around 1. I'm sorry about writing so much on the slides. The number is around 1 in a million PBNCs. And if you measure HIV DNA, you're measuring a different thing, but you're measuring the total amount of HIV DNA, you get a number that is probably 100 times higher. And if you look at integrated DNA, you get a number that is also around 100 times larger than the viral outgrowth assay. And the truth is probably somewhere in between these two numbers. This doesn't represent the whole reservoir. This doesn't represent replication competent reservoir. We know that when you look at the HIV DNA, so you're looking at viruses that are sitting in the chromosomes of the patients. This is work done by Bob Celicano. I don't know why the reference is not there. When you do whole DNA sequences of the proviral DNA, you see that not all viruses are complete, are replication competent. I create a virus that will replicate, will pass to the next generation. When you do sequencing, you see that there are big deletions in the reservoir. So when you're measuring HIV DNA, you're measuring a lot of things that are not replication competent. They might express some proteins, but not a complete virus that can replicate and pass to the next cell and start replication. And this is a very important point that people should realize why there is this difference between the viral outgrowth assay and the total HIV DNA when they measure. This is what I'm going to talk about recent data on trying to cure HIV that has been presented over the last year. And this is the steps that people that are thinking about curing HIV that people think is going to take to eradicate or cure HIV infection independently if it's going to be functional or sterilizing cure. So it's going to take a kick to try to wake up the virus from latency. The virus integrates in chromosomes and sits there. So unless you activate in some way and starts making protein, RNA and protein, you're not going to be able to see that ping pong ball that looks exactly like the other million balls. So you want it to be painted in some way so the immune system or whatever you're going to do can see that ball. Then there are strategies to improve the killing, and then while you are doing all this, you have to protect the cells that are not infected to be infected with HIV that is coming out of the reservoir. I'm going to talk about advancing the kick in the reservoir, and there has been several studies presented regarding that. The first study was done by David Margoli's group in University of North Carolina. And what they prove is that you can give this drug, vorinostat, which is histone deacetylator, to wake up HIV from the reservoir that sits latently. What he did is he took cells from patients, 8 patients, and they gave them vorinostat, and he was able to demonstrate looking at HIV GAC copies inside, so they are looking at RNA expression, that after giving vorinostat, you would increase the number of HIV RNA copies in every single cell. So this experiment, because it was consistent across the 7 patients, was the first time that somebody demonstrated by giving a drug, you can activate the virus from latency. It doesn't mean that the cell is expressing protein. It's starting transcription. It's going from DNA to RNA. And this was the first time, and that's why it was published in Nature in 2012. People have used other drugs like romidepsin, and this is a study by a group in I think it's in Denmark, that gave romidepsin, which is another drug in the same group, histone deacetylators, and gave multiple doses to individuals. And in this study, 5 milligrams per square meter, after giving the romidepsin, they were able to see viral copies in the plasma. So this is the first study that went beyond what David Margolis had done. It's not just in the cells. You can see a complete virus that is popping out of those cells. And they did it in 3 cycles, and they were able to show consistently that they could detect viremia. They also showed that after 6 cycles, the cells somewhat became resistant to this mechanism, and that the viral loads were lower after the 3rd cycle of romidepsin. So this proved, that you have complete expression of proteins and viremia when you give one of these drugs, but it might be a fading effect in subsequent cycles. Important study. This is a study that was presented, building on that. It was presented at the International AIDS Conference in Durban this summer, and Ian commented on this study. So what they did in this study by a group in Australia, they took patients that had acute infection, so that were FIDIC 3 and 4. So these were people that had starting to have, they have viremia, and they were starting to have antibodies. So it's at the beginning of the acute infection. People that were caught very early upon the infection. Most of them is in a cohort in Thailand, but the investigators are also from Australia. So, and they were treated for 2 years with antiretrovirals, 2 years of treatment. So very aggressive treatment, very early during the infection, when they don't have a very big reservoir, and they were randomized to receive continued therapy, 5 of them. 5 of them continued the therapy, and 10 of them got vorinostat, one of those activators of the reservoir. While they were taking antiretroviral therapy, they got an anti-inflammatory hydroxychloroquine that sometimes we use for lupus and other inflammatory disease, and they got maraviroc, basically to prevent if any virus would come out with the vorinostat, because you block the infection in the next cell with maraviroc, it will prevent. It will be just expression of the virus, not completing cycles of replication. And they were monitored very quick. And then after doing this with several cycles on and off, they would stop antiretroviral therapy and follow the patients until the participants, not patients, because they're people doing well, until the viral load was over 1,000. And they wanted to see how many people would not rebound, because they would have eliminated the reservoir. This activation would eliminate the reservoir. That was the hypothesis. And the results were very sobering. Everybody rebounded very quickly, 2 to 4 weeks after stopping the therapy. And in fact, the use of vorinostat with antiretroviral therapy, with maraviroc and hydroxychloroquine, didn't make absolutely any difference. So we are not going to clear the reservoir just by activating it. That doesn't clear the reservoir of the virus. If your measurement to the clearance of the reservoir is it takes longer to the virus to rebound when you stop antiretroviral therapy. The only patient that stayed a little bit longer with an undetectable viral load was one of the controls. This is not the type of result that you want to see when you do this type of study. But it shows how difficult that these things in isolation, just activating the reservoir with the idea that you will somewhat delay the rebound of viremia, is not going to be enough. So what have we learned over the last year or so about kicking the reservoir? That you can activate the reservoir using some of these histone deacetylators like vorinostat. There is other studies looking at panovinostat, romidepsin. There is other molecules also that people are using to try to activate HIV reservoir that increases HIV RNA expression intracellularly. It can be associated with transit viremia. Subsequent treatments might be less effective, so there is a compensation mechanism. If it does alone, and I think the trial in acutely infected shows that, does not eliminate the reservoir, so that not might be the solution to eradicate the virus. So now we are that's the kicking. What is new about eliminating the reservoir? So people have used different strategies to try to eliminate the reservoir. In a specific, as I say, kill everybody. So you have one ball out of 1 million infected. If you eliminate 1 million balls, that guy is dead. That's what was done with Timothy Brown. I know it sounds extreme, but basically what they did is very aggressive chemotherapy, removed completely his immune system, his own immune system, and gave him an immune system, new immune system with a bone marrow transplant that could not replicate. So, and then he developed graft versus host disease. If there was any cell left from his original immune system that might have some reservoir in it, it was eliminated by the graft that he got. So that's a pretty aggressive movement. We are looking also at drugs like kill everything, like ACTG, antithymocytic gamma globulin, and we are looking at effects on the reservoir in people that get antithymocytic gamma globulin in HIV positive renal transplants to prevent rejection, but we think that drug might have an effect on the size of the reservoir. And then people are developing specific approaches to kill the reservoir. And we and everybody has been playing with the 3 branches of the immune system. So some people are boosting the cellular immunity with T-cell vaccines, boosting the pre-existing immune responses of the individual by blocking some of the exhausted phenotype of some of the CD4, CD8 cells, so by blocking PD-1 and PD-L1 pathway, and gene therapy. Engineering the T-cell response. People are working also improving innate immunity and also using neutralizing antibodies to try to target the HIV reservoir. And we have been doing many of these things here at Penn, and I will talk about data that have been presented recently and the kind of things that we are trying to do here at the University of Pennsylvania. So in a specific killing, the Boston cases, this is Henrik, when he talked a couple of years ago in Kuala Lumpur about patients with bone marrow transplants that had HIV and basically very aggressive chemotherapy plus a bone marrow transplant, so donor. This was not like Timothy Brown. These were people that were wild type in the co-receptor. So the aggressive chemotherapy plus the bone marrow transplant decreases the size of your reservoir. So they look at the reservoir different ways. They look at total DNA. They look at the viral outgrowth assay in patient A and B, and they couldn't detect any reservoir in these 2 patients. So they got permission to stop therapy in those patients, and both of them rebounded. One took 3 months, and the other one took around 6 months. So reducing the size of the reservoir maybe delays the rebound of the virus. But in this case, both patients had a little bit of graft versus host disease. They had chemotherapy. They had an immune system that was susceptible to HIV, so they have reinfection, basically very high viral load titers. When you look at the viral titers, the patients had 1 million copies. And it took a longer period of time, showing that delay of the rebounding of the virus might be a marker of effects on the reservoir, a good surrogate marker of the size of your intervention on the reservoir. But it shows you also that by killing everything, I mean, only 1 patient has been cured by killing everything. So in the same meeting in Durban, they presented this cohort. EpiStem is a cohort that is looking at patients with HIV that needed a stem cell transplantation because they had a disease that required a transplantation. And the goal of this study is to understand the biological mechanisms of the reservoir, the maintenance on the reservoir, and how can you reduce or eradicate it using an allogenic stem cell transplantation. This is not the solution for everybody for curing 40 million people with HIV, but at least I think using these patients, we'll understand better what is the effect of these very aggressive measures to reduce the size of the reservoir. They have enrolled 24 people so far, and they presented data on 3 patients there that look at, they receive bone marrow transplants from people that were wild type or Delta 32s, and yes, allogenic bone marrow transplant reduced the size of the reservoir. independent on how you look at it. If you want a single copy assay, if you look at the HIV DNA copies or the Quiboa, you see a dramatic reduction on the size of the reservoir by an allogenic bone marrow transplant. This cohort, I am sure that we're going to hear a lot about this cohort in the future. People have tried to reproduce Timothy Brown. So Timothy Brown is not the only individual that has had a bone marrow transplant with delta 32 donors. The results of that has not been that good. So if you look, this is Timothy Brown. If you look at the 7 patients that have been done, that have been presented, all of them died. And they died because of the underlying disease, not because of the, particularly for the bone marrow that was used with the goal of trying to cure or eradicate HIV infection. But this data tells you that curing HIV in a significant amount of people with a bone marrow transplant, even if it's a delta 32 donor, is not going to be the solution for the cure of HIV. So we have learned from these cases that it's possible to cure HIV, that is very difficult, that very, very aggressive strategies to remove the reservoir, including total body radiation, chemo, allo-BNT, ATG, with graft versus host disease, it doesn't get worse than that, do not eliminate the reservoir. And treating very early, neither. So we probably will have to settle for a functional cure, not an sterilizing cure, at least as a first step to a cure of HIV. So killing, eliminating the reservoir. What are people doing to try to eliminate the reservoir? We talk about, and we are going to talk about now about specific killing, not in a specific killing like in a bone marrow transplant. This is a very important paper that literally was published yesterday or the day before yesterday. The study was presented at CROI this year, and they started to use combination. So in this study, try to boost the immune system of the individual and then combine with an activator of the reservoir. This is the first study that has done a combination approach to cure HIV. So they use a vaccine. It's the same group that presented the romidepsin study. It's from, I think it's Denmark, but I am not sure. So they use a vaccine that tries to immunize the patients, and then they use romidepsin with an adjuvant, GM-CSF. And then they use romidepsin to activate the reservoir. So what they did is, they vaccinated patients that were doing well on antiretroviral therapy, and then after the vaccination with the adjuvant, they gave them romidepsin. So they want to boost the immune system and then activate HIV. So they want to create a CTL response against HIV before the ping pong ball becomes red and the immune system can find it. So and they gave romidepsin, and then after a while, they did an analytical treatment interruption. They measured the reservoir by very sophisticated methods, IUPM, before, after the immunization phase, and after the activation phase to see what is the effect of this combination in the reservoir. They look at HIV DNA and integrated HIV DNA. And what happened was, during the vaccination, not surprisingly, there was no difference in the size of the HIV DNA, that's the size of the HIV reservoir. After that, they gave the romidepsin, the 3 cycles of romidepsin, and they saw a decrease on the total HIV DNA. So there was a decrease in the total HIV DNA in the post-activation phase. And when they look at IUPM, so the infectious particles per million CD4 cells, there was a decrease of the total number of the reservoir of around 40%. And this was consistent among participants. You can see the individual curves here. They stopped therapy, and the same thing. Everybody rebounded. This was around 20 patients very, very quickly. So vaccinating people to try to create a T-cell response, plus vorinostat to try to reactivate the virus from latency, although decreased the size of the reservoir by 40%, didn't prevent the virus from coming out at all, or delay the rebound of viremia. And this is the rebound of viremia, and this is the number of days. And as you can see, this is, if you compare to historical controls, this is exactly the same kinetics that you would expect if you didn't do anything. So the results are sobering, but this is the pathway. This type of design is what is going to be used in the future, combining approaches to try to have an impact on the reservoir and measuring the impact using treatment interruption, which is a model that we have used here at Penn for many years now. Sure. One of the patients had a delay. This is the delay of rebound. I think you can, Una is asking that maybe some people have a better benefit, a bigger benefit, so they have a big decrease. I mean, it's true, some people you might have a bigger decrease in the reservoir, but it's very risky when you enroll a cohort of individuals. You're going to have a normal distribution, and if you're picking the ones that look like the ones that you like, it's going to be very difficult to interpret. So we really need to look at the whole population and what is the impact in the reservoir in the whole population. But I think you have to be careful. It's fun when you look at some people interpreting some clinical studies, they look at responders and non-responders, and there is a statistically significant difference. Yes, sure. If you divide. It can be, I agree. I mean, it could be, I agree with you, it could be biological or it can be random. So, and that's the key, is to try to study those individuals that are outliers. You can learn a lot from outliers, and, but it could be a random phenomenon. So then you can engineer the kill. If the vaccines don't create the T cells that you're going to need to eliminate HIV, you can engineer the kill. The way CD8 cells recognize tumor cells, in this case, or HIV-infected cells, this thing move, is by the presentation. So the cell that is infected or is a tumor cell presents a peptide in combination with MHC class 1, and the T cell receptor recognize that as something with a co-signal, that something that has to be killed, and sends perforin and other things to try to eliminate the tumor cell or the HIV-infected cell. And you can clone and create those CD8 cells by putting them specific T cell receptors, and this is done by a company called Adaptimmune, and we had a collaboration, and we started a clinical trial looking at this approach. Or you can engineer something that looks like a T cell receptor, but it's called a chimeric antigen receptor, that you can design the specificity. It will basically make the CD8 cell, you put the tail. So this is an antibody or a molecule that will bind to a tumor cell or an HIV-infected cell. Has been very successful in the treatment of leukemias using a CD19 antibody. And then you put the tail that has all the signaling that will activate that cell to kill the tumor cell or the HIV-infected cell. And here at Penn, Jim Riley has led the use of chimeric antigen receptors to be used in curing HIV infection. This is a trial. Adaptimmune had this technology that it can clone a T cell receptor that was from a long-term non-progressors into CD8 cells. And we did a study using gene therapy. We collected CD8 cells, CD4 and CD8, but we used CD8 cells. We put the new T cell receptor that was specific, a very strong response against HIV. We match with the right MHC class A2 in our case, and then we reinfuse them back into the patients, and 1 week after that, we stop therapy. We enrolled 2 patients. We have never published this study, by the way, or presented in public in a meeting, and we gave a low dose, and this is the total dose that we used, 20 million of these transduced cells, or 65 million. And what happened is the patients rebounded, some of them a little bit later than expected, and the CD4 count went down, and we were getting ready to use a higher dose in this study, and the study was shut down by us in joint decision with the NIH, which was the sponsor. And the reason why it was shut down is in a different study that was done to treat using T-cell receptors to recognizing this antigen, which is from a melanoma cell, from melanoma. In a different clinical trial that used gene therapy using T-cell receptors, there were 2 deaths, early deaths, with myocarditis. And so there was questions about the safety of this approach. No, our patient, I mean, the T-cell receptor that we used had been in a human before, so it was not risk, but based on this study, NIH and we decided to stop the trial. And the reason why this happened, and this is to the credit of Carl Jung, that really characterized why it happened, is because this chimeric antigen receptor recognized this protein, titin, that is only expressed in the beating heart. So when they screened this T-cell receptor, they didn't see that it was cross-reactive to anything that was alive, but it produced myocarditis because this T-cell receptor not only binded to the melanoma, to this antigen, MAH3, or it binded to titin too, and that's why it produced myocarditis. It was a very sobering experience, and I agree that we had to stop the trial because most of our patients are doing very well, and the risk-benefit balance is not there. So you can engineer cells to become using chimeric antigen receptors, and there has been several generations. You put a tail end inside the cell that is the signal for generating cytotoxicity, and then you engineer the outside of the cell to recognize what you want. You can put an antibody, you can put another protein that will bind to something, and then over the years there have been modifications in the tail end with CD28 that increase proliferation, and Jim Riley can give a talk about all these things that you put in the tail and the difference between them, and you can modify and use other signal in the tail, in the intracellular different domains that will increase the survival of these engineered cells. This has been used before, many years ago. These are papers from 2000s using a chimeric antigen receptor in CD4 and CD8 cells that express CD4. So you can, outside you put CD4, inside you put the signal, the CD4 SIDA, to activate those cells against HIV-infected cells. It was given to patients during acute infection and also chronically infected patients. Steve Dix led this trial, and I think Romit Sayasu did the other trial. And the good news is that there was no off-target effects. The bad news is that there was not a lot of antiviral activity in these cases, although there was a small signal there that suggested that they might have some antiviral activity. The other good news is that when you do this, you can follow these patients for many, many years, and these cells, probably by homeostatic proliferation, they are still present 10 years after they have been infused. So these patients came every year for 11 years, and still they were able to detect these genetically modified cells in concentrations that were maybe 1 in 1,000 in the periphery of these patients over 10 years, and nothing bad happened to the patients. The patients were on antiretroviral therapy, so they were not killing anything because there was no expression of HIV proteins, but the cells survived for 10 years. So this is a signal that you can do this, and you are going to have potentially, you have built an immune response that is going to be long-lasting. Now with the development of new tail ends, and you can engineer cells for more cytotoxicity, for more durability, and Jim is working on all that, looking at the design of the new CAR T cells that we will be using in subsequent clinical trials. So basically that's the objective of one of the objectives of the Delaney collaborative that Luis Montaner and Jim Riley direct here at Penn, and we want to do a clinical trial that is using chimeric antigen receptors to combine approaches. So we want to make chimeric CD8s and CD4s that are targeting HIV by using CD4 outside. We want to make those cells resistant to HIV because if you put CD4, those cells become susceptible to HIV by maybe removing, as we have done in the past, the co-receptor, and then we use it in patients. So our hypothesis is that these engineered CAR T cells with an activator of the HIV reservoir will be safe, well tolerated, and will delay or prevent the return of HIV viremia, and it will be associated with a decrease of the HIV reservoir. This has been funded, and we are in the first year, in the first month, literally, of this grant, so we will be doing the studies. This is the project that we want to do. We will manufacture cells. We will give them the CARs with the chimeric antigen receptors. Here, we will look at the effects on the reservoir at each step of the way. Then we will give the activator of the reservoir, and then we will do an analytical treatment interruption to see if delays the return of viremia, and then we will restart therapy in everybody. So this is a schema of the trial, and then this is the different endpoints that when we wrote the grant, we were thinking in doing. It's a very intensive study. It includes several leukapheresis and multiple rectal biopsies to make sure that these cells traffic and that the effects on the gut lymphoid tissue. You can try to boost the immune system instead of engineering the T-cell response. You can try to basically awake response that was existing, that it was exhausted. And in cancer, they are doing this to try to use the PD-1, PD-L1. PD-1 is a marker of exhaustion in T cells, and when they have been engaged, and this has been demonstrated in chronic viral infection in the mouse model, when they start expressing PD-1, they become less effective. So if you give an antibody that blocks this axis, then those cells become more active again against the target. This has been used in the treatment of cancer for melanoma very successfully. And there has been many papers in the last 2 years in the New England Journal looking at that. But you can do that also to see if by blocking PD-1, you can reinvigorate the T-cell response against HIV. Some critics would say, well, the virus already mutated around that T-cell response, so you are just basically increasing something that is not very effective against that particular HIV infection. But I think still we want to see if you can improve in the immune response that the participants have. We did a trial here at the University of Pennsylvania and the ACTG group looking at this drug, BMS 936-559, different dose. This is a PD-L1 antagonist, and we enrolled the first cohort. And the results were presented by Joe Aaron at CROI this year of this first cohort. The study was stopped because in animal models, it was toxic for the retina, something that has not been seen in any of the human trials in patients with cancer or in patients with HIV. There was a moderate improvement in some participants, not all of them, and this is the issue about how it's very difficult to interpret when you have only 8 patients. There was some moderate improvement of immune response against HIV, some T-cell response that were increased, but when you look at 8, some of the patients didn't have improvements or even decreases, so you don't know how much is random or is real. And one of the participants hypopituitarism, adrenal insufficiency. The problem with reactivating and giving these drugs, off-target effects. You develop autoimmune responses against your normal tissue. This is something that happens in patients with cancer, and it's something that you don't want to happen in patients that are doing well taking 1 pill a day. So it's different if you have a melanoma and you know that unless something works, you're going to be dead in a few months. It's very different, the balance between risk and benefit on somebody that is doing well taking a single tablet. So this shut down the study, and I think it put a break in the whole idea of using checkpoint inhibitors as a way to boost the immune response in patients with HIV. It's going to be difficult to put a study like that through IRBs, I think. But maybe new checkpoint inhibitors will be more specific and will not generate this type of off-target effects. Improving the innate immune system is something that mainly has been pursued by Luis Montaner. This is a trial that was published 2 years ago. He looked at the effects of interferon on NK cell function, interferon boost NK cell function, and to see if that would prevent the rebound of viremia after stopping therapy. So the usual model is antiretroviral therapy. The patients were randomized to get relatively high, normal dose or half dose of interferon, and over time, off antiretroviral therapy, see how many of them will rebound, and then everybody goes restarted on antiretroviral therapy. And what happened is here. This is the historical controls. This is the amount of people that rebounded. So we know that almost everybody rebounds by week 8 of stopping therapy. In the people that receive interferon, half of them didn't rebound for 12 or 24 weeks. And these results was pretty amazing. In fact, I remember, and I tell always the story, that when people had been suppressed for 12 weeks, I told Luis, I think these patients are taking the antiretrovirals on the side. Because you always have to look for trivial explanations. They were not. I mean, we look at levels and they were not taking. So interferon alone controls the rebound of viremia and is associated when they look at the size of the reservoir by using UNA assay, integrated HIV DNA in CD4 cells. They saw a decrease in the people that receive interferon. And this is the base, and there was people that had, it's curious, but people that rebounded had a bigger effect on the size of the reservoir. This is the base for the other big project in the Delaney collaborative, looking at interferon in combination with broadly neutralizing antibodies to see if we can reproduce this and increase the effectiveness using broadly neutralizing antibodies. This is a study that we're doing now, try to characterize better, very similar to the previous one, looking at interferon during an analytical treatment interruption, interferon with antiretroviral therapy. It might be that you don't need to do the analytical treatment interruption to decrease the size of the reservoir, and a control group. The study is enrolled, and I think it has enrolled around 2/3 of the participants. And then we have talked about innate immunity. We have talked about specific building T cells, specific immunity to try to eliminate the reservoir, and then people are working using the humoral response, usually using broadly neutralizing antibodies. That has been on papers over the last 2 years. So we did a trial using one, there is a slide missing here, but we did a trial using BRCO1. So these broadly neutralizing antibodies bind to different parts of the GP120 molecule, the CD4 side and also stem and many different areas, and there was a slide explaining the different areas that these antibodies bind. These antibodies are made by individuals with HIV. They neutralize many different HIV viruses in the community. They don't work for their own virus, but some of them have been characterized very well, and they have been manufactured to be used as drugs and both in prevention and in therapeutics. So BRCO1 is one of these CD4 binding antibodies, and this is a study that we did with ACTG. Katie Barr and I, we were the co-chairs, and we gave multiple dose, very high doses of BRCO1 to patients that were fully suppressed, and then we stopped therapy, and we look at what happened. Our hypothesis was that by using very high levels of BRCO1, it would prevent the rebound of viremia. And we look also, the dose that we used was 40 milligrams per kilo, and we gave it 1 week, just 1 week before stopping antiretroviral therapy, and we monitor viral load every week. And something that is already a theme here, everybody rebounded very early. Some people, it took a longer time to rebound. 2 individuals, one rebounded at week 11, and the other one rebounded at week 8. So there was only a slight delay in the rebound. So when we compare what we saw to historical controls, there was a statistically significant difference at week 4 of the analytical treatment interruption. So more people have not rebound at week 4 than expected by historical controls. And at week 8, the amount of rebound was exactly the same. So there was a delay on the rebound of the HIV, but not very significant. And this has been accepted last week as a publication in the New England Journal of Medicine, and hopefully in 2 or 3 months we'll see a printout of this. It's going to be published in combination with a similar study that was done at the NIH, a little bit smaller in size. Other groups are thinking about the same lines. So this is a study that has already been published in Nature by Marina Kasky and Michelle Nuisensack and Slade from the Rockefeller Institute. And what they do is they use a different antibody, 3B-NC117, and during an analytical treatment interruption. Similar design. So they gave the antibody, 2 doses or 4 doses, and the patients stop therapy and they follow for the amount of rebound. And here is a very complicated slide, but this is the most important figure in this figure. So in this paper. So when you look at the historical controls in black, this is the rate of rebound. So by week 6, everybody has rebounded. When you look at people that got 3B-NC117, there was a delay that was greater than in the VRC01 study, suggesting that this antibody is more potent than the VRC01 in delaying the rebound of viremia. And this was published a couple of months ago in Nature and is an important study. So for the Delaney, we're going to be asking a combination of antibodies, some binding to the CD4, some binding to the stem, 10, 10, 7, 40, and we are combining it with interferon to see if that will prevent the rebound of viremia, if we can reproduce this data, and those studies will be done hopefully next year. Then the last step is to protect the uninfected cell during all this process. This is the paper that we published 2 years ago looking at removing CCR5 in autologous CD4 cells in people infected with HIV. You know the study, you use zinc finger nuclease to knock out CCR5 from the cells of the patient. You deliver this using an adenovirus vector. You collect cells with apheresis. In tissue, you infect them with the adenovirus, and they express this plasmid that has the 2 zinc finger nuclease and removes in a proportion of those cells, removes the co-receptor, and then you infuse them back into the patient. This is the design of the study. We have presented this many, many times, and we stop therapy. We wait 4 weeks before stopping therapy, and what happened was that everybody rebounded. 1 patient controlled viremia in the absence of antiretroviral therapy during the treatment interruption, and then everybody restarted therapy, and everybody became undetectable afterwards. And this is the reason why it was accepted as a publication, is that there was a difference in the survival of cells depending if they have the genetic modification or not. So the CD4 cells during the analytical treatment interruption that had been modified and didn't have CCR5 survived more, and this was statistically significant, than those cells that had not been genetically modified in the individual. And because of that, it proves that by removing the co-receptor in autologous CD4 cells, you prevent the death from HIV, probably from direct HIV infection. We're doing a trial, and next week we will enroll the last patient in the first 2 cohorts. In these cohorts will be fully enrolled using a different delivery system to deliver the zinc finger nuclease using a MaxCyte machine. You use mRNA to deliver the zinc finger nuclease. And we use cyclophosphamide to try to increase the engraftment, to increase the dose of the genetically modified cells. The study is ongoing, and so far it's looking interesting. We'll see the results when all the patients have completed the analytical treatment interruption. We are looking for Delta 2 heterozygous and Delta 32 heterozygous, and these patients are difficult to find in an area of the country where, because of the demographics of HIV epidemic in Philadelphia, we don't have that many Delta 32 heterozygous. This approach of protecting the cells protects only for R5 tropic virus. It doesn't work for X4 tropic virus. If they are not using the co-receptor CCR5, the approach will not work. And then Jim Hoxie here had the idea of cloning, this is not C36, C34 is a peptide into the co-receptor that blocks HIV. This binds to the GP41 and prevents the fusion of the virus into the cell. So he cloned that peptide, similar to T20, a little bit longer, 34 amino acids instead of 20, and it makes that virus, that cell resistant to HIV infection. He cloned it in different co-receptors, but it turns out that when you clone that peptide in C34 peptide in CXCR4, is more active, more effective than even if you clone it in CCR5. And it works for both CCR5 and X4 tropic virus. So this is co-culture studies, and I am running out of time, so I want to leave a few minutes for some questions. And it's effective against both viruses. We have been approved to run a study looking at this. This is the design of the study, and the study hopefully will be dosing the first patient before the end of the year here at the University of Pennsylvania. It's currently under review at the IRB. It has been approved by the FDA. That's where we are with it. To do these studies, it takes a family. I am just the clinical trialist. I do the clinical research, and this also helps with the designs of the clinical trials. But it takes a work of many, many people. Carl led the effort at the beginning. Now Jim Riley is working on the chimeric antigen receptor and try to find which is the best one to try to target the HIV reservoir. It takes a lot of people in the clinical trials unit. Over the years, many nurses have worked with these participants. Joe Quinn, Rob Roy has been a co-investigator. The ACTG, Dr. Frank is a co-investigator in many of these studies. We cannot do this without the support of the Center for AIDS Research and our collaborators in industry. NIH pays the bills, and we do all of our studies in the CTRC, and we use the infrastructure of the ACTG to make this possible. And that's all I wanted to say. So it's possible to eradicate HIV. People are looking at combinations approaches now. Many different groups have looked at kicking, killing, protecting, separated. Now we are in a new era. We're combining these approaches. And hopefully we will be able to delay, as a proof of concept, the rebound of viremia using some of these combinations, and that's where we are now, and that's going to be the agenda for the next 2 or 3 years. And thank you very much.